to 2 meters high, and they are just lined with these very big heterocone.
Yeah, yeah.
Seeing a lot of different fish too than we have been seeing at this shallower site.
Yeah, might get. This is like a stripy red-banded rockfish, perhaps.
Mm.
Yeah.
So for folks who have access to the internet, there are the previous dive that Abby was talking about, I think was NA121 was the expedition. So if you want to check the archives, you may be able to dig up some of those photos of the glass sponge reef she's talking about.
Really worthwhile. Well, not quite
far.
reefs,
Like,
but
tug it on me. Maybe.
definitely
Nope,
like an important sponge ground, dense aggregations of these sponges.
A concentration, shall we say.
Yeah, yeah.
There you are.
I think a reef would be, you know, you want to see sponges growing on generations of previous sponge skeletons, which didn't seem to be what was happening in the situation at Gray's.
There we go.
Yeah, and that's a really good distinction, and a reminder that we need to be careful with our language. I shouldn't have said a sponge reef because that actually has a very specific definition. So thank you, Abby, for pointing that out. I think it's a good reminder.
Yeah, why don't you just come down to where Argus is? So if you go north right here, and then just kind of we'll go down as far as we can and see if they want to back up. Yeah.
About how we talk about and characterize ecosystems.
Yeah, and it's definitely still a very pretty unique environment. That's the thing about these sponge grounds are not that common, but you really do find in a few places and different parts of the world, just really unusually high concentrations of particular types of sponges that create these unique environments. So kind of lucky to have a glass sponge ground off the peninsula.
And that wall that we were, while we were exploring yesterday was pretty incredible too.
There we go.
Yeah. Yeah, a very different complement of species as well. Yeah.
Okay, science, so we are at 400 meters, we're slightly north and upslope of our target 3.02. Going back to it, we could backtrack to it and start there, or we can start here, it's up to you, and check out what we got and move along.
What's the slope and environment look like here? Are there any steep areas nearby?
It looked like it when we came down. Well, so first we're going to do a little bit of ROV setup, but just talking through the plan. So it looks like, yeah, where those sponges were, we've got some varying relief. We have this kind of slope in front of us here, and then it looked like we had some more steep stuff, some walls. We're looking at in sonar as well. So it's kind of a mixed bag, it looks like.
Well, I think it's all fair game. Abby, would you prefer to go back to that little cluster that we saw once we're set up, or continue on?
Hang on, I don't seem to have HIPAC at the moment.
That cluster is actually on our way towards 3.03. We came down a bit since we saw that. So.
Would it be possible to get HIPAC up on one of the channels?
Okay, I should be able to zoom in there.
Thanks, Dan.
You copy that, Marley?
Say again?
Yeah, stand by, science, stand by.
Okay, roger.
Want to get bottom numbers?
Stand by. I need one thing at a time here. We're on the wall,
yeah,
so.
Camera's about to go black.
Roger.
All right, we're good.
Thank you. there was a request, Marley, to get HIPAC on
Yeah.
satellite feed.
Yep.
Okay, Mike, you can do your thing. Thank you for waiting.
It's nice to have a window into that nice new van on the ship,
Mm.
but
which one?
HIPAC will be helpful too.
Which camera?
It's in the back over here, I think. It's this one.
Oh, I just look like
We
a
have
fool.
our top scientists all over it.
Looking good. Thank you for the HIPAC.
Ah, got it.
Doing a quick DVL reset. All right, you're good.
These promontories are likely our good friends here.
Yeah, I mean, I think given that we're in exploration mode and we've kind of landed on some hard substrate, it makes sense to, yeah, have a look around here.
And we're right at the
And
bottom of a significant wall, which we saw.
I think we should just carry on. So I don't think we're going to be
Mhm.
disappointed either way, but I think we're set up to move east. How's the wind blowing down there
Wind's okay.
for
Oh,
Viz?
sorry, you meant down there.
Yeah, it seems east seems like
The
we'll
water.
see what happens here, but typically it's been good for the last couple dives. So there was some giant sponges on this cliff here. We glanced
Yeah.
on the way down. I don't know if you saw them there, but
Different looking than the ones we've been seeing. And a lot of rockfish
Yep.
and sablefish and sole.
Yeah, we're much shallower here than we were about on the last dive. Last dive we were 1,000 meters and 1,100 meters, and here we're about 400.
Are these still the solid sea cucumbers? Looks like it.
yes.
That really looks neon yellow. Is it really neon yellow?
It seems that way.
You want a 10-meter bump towards you, just to
Could we maybe zoom on that
get a bit
yellow?
Yeah,
Yellow?
yeah,
thing?
Roger, coming down.
Thank you.
bridge nav.
Yeah, Roger that. Right. Hey, I guess we'll be all right if we come up.
I'll just do 10 meters. Will be all right.
So there are two
Okay,
types
Marley,
of
you
sponges
can
that we
zoom in
commonly
there.
see.
Bridge nav.
That are bright yellow like this. This might be a type of yellow staurocalyptus that's just got crinoids, ophiuroids hanging out on the surface, using it as a perch. But one thing we've been finding with these samples of these yellow staurocalyptus is that once you get them on the ship, it's almost like the yellow just washes out of the sponge tissues. So it'd be kind of interesting to get a chemist to figure out what's going on, whether the color is coming from like a symbiont, or whether it's some kind of metabolite the sponge is producing that then easily kind of dissociates from the tissue.
It's a really pretty shot.
Nice.
Yeah,
Yeah, with that rockfish that just wiggled in
Yeah.
there.
beautiful.
Is that Swiftia too?
I think so.
Fan? I don't look so red though. I don't know. Maybe I'm wrong.
Thank you for the zoom.
Yeah, we can hold that for a minute. I'm just playing with my trims, so.
Can I zoom in a little further?
Yeah, you can zoom right in. Give me a chance to dial the trims in here, so the vehicle will hover without input from the pilot.
Yeah, thank you, Marley. It's really helpful to get that detail. Are there patterns to the openings that you see on the sponge surface? Is that one of the diagnostic tools you use, Abby, or how do you?
So with this particular type of sponge, you look at, there's a whole load of things that you look at. You look at the kind of the gross morphology of the sponge, then with the glass sponges, the inner and outer surfaces can be quite different. And like sometimes you'll find there's like a thin lattice or mesh of a particular type of spicules that will cover all the little pores on either the inside or the outside, and that can sort of give you a clue. But with these, for instance, to tell a Staurocalyptus, you have to look at, they have these little spicules on the outer surface that kind of look like umbrellas without the fabric. And you, under the microscope, you can look, and some of these have thorns and some of them don't have thorns. And so it's not until you really start looking at the spicules under the microscope that you can start to sort of pick out which genus a sponge might belong in. So this one I would say might be one of the yellow Staurocalyptus, which are on the collections list for, and it's been requested by a few different agencies. So it would be one to potentially take a sample of. But again, I think they're likely to be quite common, so I think we'll come across more of these.
Yeah, and that's a good note too, is trying to find samples that are easily accessible by the tools that we have. Primarily, we've had success with both the manipulator arm and the slurp sampler so far. So it's always interesting to know what sort of textures these sponges might have, which helps us decide how we might want to try and grab them.
Yeah, so sponges have this the sort of equivalent to a skeleton is made of glass spicules, and these particular sponges that we're looking at now have kind of a loose framework, but it almost has a kind of fiberglass-like texture when you get it on the surface. So they tend to sort of tear if you're taking a sample off. Yeah, contrasts a lot with some of the little demo sponges. Yeah.
Yeah, and that's part of why we have to keep all of the sponge samples separate, because we need to know which spicules go with which samples.
Yeah, and it really can be quite a nightmare afterwards, just because most sponges you'll get some spicules that are very common, and as soon as you take a tiny sample, you'll find lots of them. But sometimes the diagnostic ones that will tell you if it's a particular species are really, really teeny tiny, and there might not be so many of them. So you really want to know that what you're looking at came from the sponge you're trying to identify. So yeah, if you had two getting mixed together, that could really throw you off. And yeah. So like you say, Jenny, it's pretty important.
I'm all the way zoomed right now. Yeah.
You can come out if you need to, Marley.
Dan, where are we at?
Oh, I was just trying to see inside, see who's home. That's a good spot for a sample if you want, or we can move on.
What do you think, Jenny?
Abby, that's up to you. Yeah, let's have a little look around.
Yeah.
Okay, you can zoom out, please. Thank you. Oh, I got far away. No wonder.
There's a lot of water in the way.
Oh.
Seems like the rockfish are interested in us. Erica, are you on? Are they sharp chins? What kind of rockfish are these?
So I was looking at my notes from the last analysis we did from here, and we saw green striped rockfish, flag rockfish, sharp chins, and they're the ones with the dark V behind the eye, and rose thorn rockfish. They were the most common ones that we came across last time, so chances are we'll see some of those again.
Thanks, Abby.
Yeah. What else?
I'll move a little closer, Rennie, and I'll do the other side of this little output here.
Right, I think I can step north and kind of get on the other side of it.
I'll just move closer, mate.
All right, so that'll be 045.
There's so many
Great
crinoids.
stuff.
I know.
Yeah,
all
big
right
Only
ones.
on that one
of the
side
feature.
though, yeah.
One
Mhm.
step,
Just 10 meters.
10 meters, 045.
Then we'll move north.
Wow.
It curves around.
There you go.
This little output here. Out. What do you call it? Peninsula.
You know, I think it's really interesting that they're all clustered here.
Mhm.
As we know, crinoids can swim and sort of walk and move, and it tells me that they may have all grouped themselves here because it's really good feeding, which makes me wonder what else we're going to find.
Yeah, most likely the little sticky
It's,
arms catching all particles.
It's a bit of a little peninsula here. Then it goes for about 10 meters and then dog legs.
Something tells me we're going to see a lot of
A
those.
90-degree dog leg here coming up, and
Neat little features and promontories all along this slope. I love following them, just like this.
Holothurians and crinoids seems
come
to be
down
here.
5, would you, Mike?
Copy.
I've been looking for orange cup corals for some of our
That's a cup
collaborators from university. Oh, yeah. Well, that's
Just
the
under the laser.
Desmophyllum, right?
Yeah.
Yeah.
Yeah, I think so.
I think they were looking for Balanophilia, which I don't think we've seen yet. Lots of
my
spot
coral list up.
ponds.
Kind of where the two walls meet here. We're in a little, it's like the walls are 45 on each side of us there with a little creek in the middle.
I would expect these little chutes to have more sediment, but it does look like there's some nice rocky habitat here.
Heteropolypus.
This is
Yeah.
yeah.
You want to zoom on him? Zoom in for a sec, Miley. This is somebody's favorite, I forget. You can zoom in a little more.
So funky.
Looks like a dog toy.
Yeah.
It
Totally.
does.
A mushroom coral. They do have a wonderful Latin name though, Heteropolypus. I mean, pretty good.
Okay, you can zoom out. A little close there. Ernie, can you change the
You want 5?
mesotech to 5 meters, please? Yeah.
Ooh, lower right. That's a nice heterocone.
Yeah. How'd you
Oh,
go?
and upper right. Another big one.
Come up just a couple meters, Mike.
The two of us together will make a full data log. Huge heterocone.
So I think we're probably seeing a mix of Heterocone calyx and also Africalystes vastus.
You wanted to
These
see
ones
this one,
down the
right?
bottom,
I think.
Heterocone. Sure, yes, please.
Is the Africalystes, is that the quesadilla sponge?
Yes. But the quesadilla, I'd say that's kind of a more unusual morphology. They grow into barrels, but see, like this
Zoom in
Heterocone.
a little bit there,
When you look at
Marley,
them
if
under
you
the
want.
microscope, the way you tell them apart is they have different, like the surfaces on the inside and the outside are different between those two species. But just looking at the morphology like this one with these big kind of roundy projections on the outside is kind of classic Heterocone.
You
Whereas
can,
the Africalystes are
zoom
kind
in
of
a little more if you want. The structure.
Dan wants to know who's home.
Yeah. Nobody's home.
Nope.
Or if they are, they're way down in there.
Now this one seems to have a similar texture on the inside and outside surface. Is that true when you have them in hand?
I can't remember off the top of my head which way is which. It's either heterocony or one of them has the same surface on each of the same texture on each surface, and one of them has different on the inside to the outside. There's also there's a little
Thought I just did.
associate
I'll do it again. Just in case.
that is only
Oh,
found
it
on
was like
heterocone. So if you see
literally-
that, then you also, that gives you a good clue.
Could you zoom out just a little bit, please?
Roger. Is that good?
Beautiful, thank you.
Okay,
All right.
Jenny, I've just got my
Beautiful.
notes, and yes, heterocone, the surfaces are the same. So the dermal and the
Interesting.
gastral, same.
All right, shall we ramble on?
Sure. Yeah, just let us know. Let you get a little further so we see the direction. I can bump 10 towards you.
I can't quite hear you, but you can see what the cliff that goes off to our
Yeah.
right
So,
there.
yeah, for that's the lower layer where Herc is, and then there's the upper where Argus is. So I think there's, yeah.
I can move. Never mind.
Yeah, it looks like I'm going to be moving east, so
All right.
if you just want to move east a little, see what happens.
East will take you off, but all right, I'll try it.
Yeah, or well, yeah, you can do an angle there, whatever angle you think's ready. But generally, it looks like our ROV is going to go east.
Okay. Bridge nav. Let's do 10 meters, 060. Thank you. So you see that light bit right in the forward of Argus? I think that's the ledge you're looking at. And then that kind of ends.
Yeah.
Yeah, right.
Compass of dead farrier, a big clump of live farrier down the bottom as well.
What was that?
So this big clump of glass sponge down in the bottom right corner.
Oh, the Freya. Oh, yeah, I'll write that.
Mm-hmm.
There's the top of that right there, I think.
Are the small pink ones more heteropolypus?
Looks like it.
Yes, I think so. Could we take a sample of the farrier, Jenny? Or pilots?
Sure. Pilots, is it accessible?
Sure.
Maybe slurp?
Yeah.
Thank you.
Which one do you want? Sorry.
The frilly white one.
Bridge
I'll
nav.
Frilly
So
white.
the lasers,
Copy.
the lasers
There you
are on
go.
the dead patch. Here we go. There's some live underneath.
Hold position.
You want to sample the fish?
Been easily
No, it's
slurped in
still
the
doing-
past, as I recall.
Yep.
Telestrator's still doing the thing where it's off by a little. So not the fish, the big sponge next to the
So
fish.
the telestrator or the telestrator operator?
It is the telestrator. No, now it's good.
Oh, now it's good.
And I'm hoping that, because there's a little swiftier coral in there as well.
Yeah.
that
Can try a
Meredith
little
might chime in.
pre-sample zoom there if you want.
Are we gonna slurp it?
Is that the red
Yeah.
sticks with
Got
the,
a slurp jar.
Fan.
Not the sticks, it's the fan.
ah,
Red fan with yellow polyps. But we might have a lot from up here because OCNMS is not in the permit regions for it, so we might not need that one.
All right, I'm ready to flush.
Thank you.
it's actually a very good point to bring up. We are working outside Olympic Coast National Marine Sanctuary, and so the requirements are for sampling are not as controlled by the permits as they would be within the sanctuary boundaries.
Yeah. It still gives us a good clue, though, that if it's not on for OCNMS, then it likely means that we have quite a few samples of that particular coral from this region.
Yeah, and we
Whereas
are
the
very
barrier,
careful
we know
not to, not to oversample. You know, if we already have samples, we work with those. We don't always just keep collecting
to
more and
flush.
more and more. We try and be quite cautious.
What's going on with that hockey
Oh,
puck?
come on.
The hockey puck was split.
That was weird.
Yeah, I think the claw got right in between them.
Don't forget the capture.
Okay. Chase it around. Is our victim. That's it.
Yeah, let me flush first.
Slurp 1 whenever you
Show
guys
me
are
the
ready
minute
flushing.
for a second.
Yeah,
We are flushing.
Rennie, this is sample 133.
Yeah, top of it.
133.
I think I'm in
Aye.
the.
Thank you.
Oh, yeah.
Okay. Yeah, good. Yeah, we're flushing. Flushing all the time.
All right. What? No, go to number 1. Is that what we want?
Yep.
That's a copy we want. Ugh.
How is there always stuff in them?
I could flush it a little. Unless you want me to go to a different one.
If flushing, it's possible.
Oh, let's let it go for a little bit.
You guys take that.
Could I get the sample number for this one, please?
Which,
Sample 133.
Oh, yeah.
Thank
I
you.
noticed that one time. The filters don't get clean.
What was that, Mike? What'd you say, Mike?
That, I was just confirming what he was saying.
Oh.
Sometimes that the mesh doesn't get cleaned off or something.
Okay, we ready?
So is this okay, or do you want me to go to a different one?
I'm trying to remember what was in one last time, and I kind of think it was the microbial mat. So, we probably don't want to go into that one, right, Science? There, it seems like the filter still had a couple, some little pieces of microbial matter still left on it. What was that?
That'd probably be all right.
That's all right?
I think that'd probably be okay. Yeah.
Okay. All right, then we'll stand in jar 1.
Copy.
Okay. Try 25%.
25.
Little crab in there too.
25% engaged.
Go
And you're
for 50.
Go for 50.
You're all about robbing the people out of their homes in these sponges. He's always like, oh, and we
Aw.
got that too. Who's
Oh
home?
no.
Bonus sample.
We're just remodeling.
Yeah,
Come out of there, you.
just kicking the kids out. It's time.
Maybe I'll leave him.
Full
He
of
gets
channels.
to live.
There you go.
Just because
He's
He'll
not our
clog up our
target
suction
anyway.
sampler.
No.
Starting to come into the jar now. Oh, I got too greedy. Go a little higher, Mike.
Give me all she's got.
Yeah, now we got some pieces coming through.
There. I can.
There you go.
Is that enough?
If that all went in the jar, I think that'll be enough.
I think there's more coming in.
Yeah.
Losing our visibility here.
Although we can't always see them, the pilots actually have more cameras that they can look through.
Turn off the jar for a second.
There are cameras that look at the jars, cameras that look at the Niskins, cameras, there's a bubble camera, camera that looks at the bio boxes on the starboard side. It's pretty amazing to see
I feel like we got more than
what
There should
images
be
they have at their disposal.
more coming through.
Yeah, all right, go back to 50. Well, they're falling down. There should be more though.
Yeah.
That's stuck on the media. There it comes.
We'll just have to do an extensive flush after this to make sure.
Yeah, I feel like there was even more than that, but all right, that's probably enough sample. Just
Well, once it's stuck on the media, you can bump it up. It's not blendering around in there. You can bump it up to 75 for a minute. There it comes.
that's probably most of it.
Yeah.
All right, so we got like 5 larger pieces or so. Is that good?
Thank you.
That sounds good.
Suction
Awesome.
off, is it?
suction is off.
Yeah, bump the jar forward and backwards. They might fall off the media.
Oh, it's coming.
Yeah, I think we're good. There you go, a little bump action. Yeah,
Oh,
there we go.
Yeah, they were all stuck.
Is that the shrimp talking?
Oh my goodness.
It's actually perfect narration, like a Pixar movie. All right, we good here then?
Yes, thank you. Yep, we don't need the Swift yet.
Oh, I guess you can put it away.
Okay. I'm getting there.
Lots of sablefish just knocking into the wall.
I wonder if our lights are blinding them. Normally it's so dark down here.
Probably.
I'll be blind for the next 2 decades.
Somebody on there. Maybe.
A quick zoom if you want while we're floating away. Kind of close to the wall. I'm just going to float.
We know you just want to see who's home.
Yeah.
Somebody? Anybody?
Anybody?
It's vacancy.
They're hiding
Everybody's been
little-
evicted.
Prime real
Oh,
estate.
man.
Okay, you can zoom out, please, and Michael, if you'll center the brow camera, please. Thank you.
Centered.
Be a little dusty coming out there.
Cool rockfish.
Into the wind.
So I think, so you can see this thing kind of ends, this eastward bit, and Argus can kind of hop over it, and then we have more terrain over on the other side.
Roger.
So I think I'll just go straight at Argus's heading. And I'll have to bump over it a little bit, but should be fine. Bridge nav.
Do these Faria look dead or just heavily sedimented somehow?
No, I think they're dead.
20
Yeah,
meters,
when they're brown.
030.
Yeah.
So sponges are pretty good at keeping themselves sediment free. So when you see them covered, see the skeletons like that, trapping sediment. Yeah. So Faria is another one of those. It's a type of sponge where it has a fused skeleton. So even once it's dead, that skeleton kind of hang around and will end up getting just covered in mud.
These crinoids really like the eastward facing walls here.
Yeah. And the Swiftia fans too.
And the Swiftia fans, yeah. And
I'm
so
looking
many
at pictures of
solids.
rockfish. Trying to figure out which ones we've got.
So yellow guy with a different
Yeah. So this
different pattern.
is probably a different type of yellow sponge that we see fairly commonly, a type of Mycale.
Oh, all right then.
Yep.
Oh, what happened?
Oh no!
We sampled the Mycale. We sampled the Mycale the other day. I think it had more of a barrel shaped morphology though, or a vase. It wasn't flat like this.
They have many.
Come back to him.
It's
I'm gonna
polymorphic.
wait for Argus.
So Abby, what mechanisms do sponges have for cleaning themselves of sediments?
Well, so first of all, like the type of morphology a sponge has can help it cope with sediment. So if you have a branching form, they're kind of a lot less prone to getting smothered as encrusting sponges. Another thing, some of them produce mucus. They can slough off their tissues. Others, I'm not sure if that's the case that's going on here, but sometimes things like sea stars or holothurians that are living on the top of the sponges, it's kind of beneficial to both because the holothurians or the sea stars are kind of feeding off particles on the sponge's surfaces, but it's also cleaning those inhalant surfaces for the sponge. So sometimes they take advantage of that. But then you have other sponges that are quite happy living buried within the sediments. There's one particular type of sponge which called polymastia sponges, and they basically, most of the sponge is buried in the sediment, but you just have their little oscula poking out, and yeah, they call them nipple sponges because you just see this little cluster of little oscula poking out from the surface of the sediment. So
but it's kind of like an iceberg in that you're only seeing just the tip?
Yep.
And there's more
exactly.
within the sediment?
Yep, under the sediment, you got the main body of the sponge. Yep. So they do pretty well in
Deceptively
sedimented
hard to
environments.
sample.
Yes. Yep. But it would be quite nice if we do spot some because we know there's a few species, but still don't quite have a handle on which are distributed.
Bring your head to the west for a minute, Mike.
Back to the west, you're right under Argus. West is towards the wall.
Look west.
Do you want to go up? Up the wall?
Well, if he looks west, we can see what's happening.
Sorry, I thought you meant move the ship west.
No, sorry.
Yeah,
Getting Mike to look west.
Gotcha.
So Kurt Whitmire is watching along, and he has identified these rockfish for us. They are dark blotched rockfish. Thank you, Kurt.
Tracks pretty well with the multi-beam up here. It's a flatter slope.
There was kind of another ledge up here that we saw on the way in.
Yeah.
We want to head east, right?
Eventually,
Yeah.
we're just facing west as we come around.
Gotcha.
Just coming
That's
coming up the hill a little
Dover
to get
soles.
out from underneath Argus, and then we'll move the whole mess to the east. $10 meters up from where we were before now.
Roger.
I think these little poachers that we're seeing on the,
Oh yeah, some little poacher fish.
yeah.
That track in the ROV is a little peninsula we came around.
Yeah.
Oh, pink urchin.
Just the one.
Just the one.
There's never just one.
I know there'll be more.
Fancy a pink urchin, do you?
No, no.
Leela wants a pink urchin.
That's
I want a
nice,
pink urchin from the sea.
Nice rockfish.
Yeah, really pretty rockfish.
So we have an AI pink urchin detector
Oh
that we can
yeah,
run on imagery. So if you want counts of pink urchins,
nice.
yeah, definitely something that you don't really want to do by hand. I think we had about 10 dives and some of them had like, I think the maximum was 7,000 in a dive.
Are you guys using a CNN?
Yeah.
Nice.
Another poacher.
I might ask you about your successes and failures with that later on. I'm thinking about doing something similar.
Definitely. Yep. We tried
You
quite
look
a few different
315 now, Michael?
approaches.
Copy.
Is that a weird basket star? What is that?
And
Oh, something dead. It's like a dead Rathbanaster,
It's a
but like
Rathbanaster.
weirdly spread out.
oh,
Yeah.
and all its legs different lengths.
Yeah.
What?
It's lost a couple in its life.
Mm.
I'm confused.
Some little encrusting
2020's
sponges.
been hard on them too.
Oh man, that's a big one.
I think I can bump north if you want.
I can come back down now to the east if I want.
Sure.
Let me get back to the north of Argus.
Yeah. I think to follow this contour, we'll be heading north-northeast as we go northeast to the next point.
Roger. What depth is that one? Is that back down at 400, is it?
No, it's about the same contour that we're on now, a little lower.
Lower. Okay.
Yeah.
Let's see if we can drop into the hole now that we're over that peninsula.
Yeah, it's a pretty cool terrain.
Also, Erica has an ID on the other sea star, Stylasterus bori. She's thrown that in the science chat.
Cool.
Oh,
Thanks, Erica.
Well, that's a rockfish.
spastic.
Some of them sideways.
Yeah.
That was a hard landing.
Should be able
I wonder
to
if
look
these
to
are
the
all
north
drop
now, Mike.
washed.
Copy.
And you can come down a bit too with me.
Copy.
Wow, that sea star that Abby just mentioned is a fish-eating sea star. Now that is ambitious. If you are a sea star and you're going to eat fish, that's cool.
So many fish. I can't believe it.
Yeah.
Why do they just keep diving straight for the slope? I'm confused.
I think they're confused.
Oh my god, there's so many.
The light, the light jacks with them.
The light and Herc's very loud.
Knock knock, who's home?
Oh, wow.
Lovely hedgehog. So here you've got a dead one.
Mm-hmm.
Yeah. And one that looks like it's partially
It's not filtering
partially
anymore.
gone.
Should be able to come down a bit more, Mike. You want to zoom on the dead one there?
Sure. Yes, please.
You can zoom in a bit, Marley.
So
Okay.
Abby, what would cause this situation? Why would the
That's good.
sponges be dying?
So this is interesting because you can see, like, to the left of, I would say, so to the left of the lasers there, you can see where there's a kind of funny white band.
Yeah.
I don't know that diseases have been studied in deep sea sponges, but I know that shallow water sponges are prone to disease or can get disease. So it could
we
be
get
something
a
to do
zoom
with
on
that.
the white band?
Yeah, I'm trying
please?
to set up. So
Yeah, thank you.
Yeah, I know that in shallow
Is
water
it-
corals there's white band disease.
Mhm.
okay,
I'd
you
say
can
that
zoom in a
typically
little more, Marley.
the biggest danger to a sponge
Come on,
would be kind of mechanical damage. So sponges that live within trawlable depth are a little kind of vulnerable to that. Sometimes they just get too big for their attachments and they can kind of topple as well. I think if feeding conditions were to change dramatically, or for instance, like the sedimentation regime got too extreme. So I was saying that sponges can adapt pretty well to that, but they can even stop pumping to prevent themselves getting completely clogged up. But that's sort of a short-term measure. They can't do that long-term. So changes to the sediment as well could be bad. But in this case, this is pretty interesting because you got the sponge is clearly partially alive, and yeah, just that white band is making me curious.
Sorry, I call those in off of SPL, but it sounds like you can hear me anyway. I apologize for that.
Yeah, that was weird. Coming from the other side.
He knows we're here.
You know, it may be those fish stirring up the sediment down there.
Yeah,
Could be.
Yep.
they know we're here.
They don't want us to take a close up.
Blame the fish.
Oh, while the sediment's clearing, Erica has got some great information about the rockfish, the dark rock.
Real quick, hey Dan, would you mind cycling the still cam?
I got it.
Or is it on already?
It's not even on. Would you like it on?
Yes, please.
Turn it on. It is on.
Can you turn the Ethernet bottle on too?
Oh, of course, yes.
Oh, thanks.
Good catch. Thank you.
So Erica was saying that is one of the species of rockfish that has been rebuilt.
It's like it clears up when we zoom out.
Yeah, rockfish in general, there are about 60 species of sebastes on the West Coast, and many of them live a very long time. And when fishing was hitting them pretty hard, it was really decimating the stocks. And over the past decade or so, many of those rockfish species have rebuilt. So, and the stocks are rebuilt. So it's a big success story. Fisheries management, really. I think yellow eye is the last one in these parts that is still rebuilding.
Yeah, Kurt Whitmire is putting more information in the chat saying that there were some pretty severe management restrictions that were put in place in response to those declines, including rockfish conservation areas up and down the coast. So after this, after the species were declared overfished in 2000, those measures went in and seem to be having a positive impact on the fish stocks. So that's a fantastic example of fisheries management.
Yeah, and spatial management tools, similar to the EFH area that we're working in. Rockfish conservation areas is another type of spatial management tool. What a beautiful little cluster this is.
Yes.
Abby, what are the ones in the center? There's like,
I was just going to say, I think that is I think that's Mycale again, but a different growth form. So it could be, it looks like it could be on a stalk, or it could just be that it's that cup shape, rather than the kind of the big open kind of bars morphology of the one that we were looking at earlier.
Area is not very happy there. Is there any sign of disease? See that sediment patch on the large heterocone? I wonder if there's any sign of disease near that sediment.
Yeah, these
I'm not
heterocone
seeing
look
the
pretty
same.
good. It was interesting on that last one. I'm going to be looking out for that white band.
Yeah.
But these look pretty healthy.
It may have cleared back there by now. We can go back and have a look if you want.
I think it would be interesting if it's not too far and not
No,
a pain. Then
it's not too far. I know where it is.
Yeah, so here we've got Mycales, a demo sponge, and then these gorgeous, huge heterocone glass sponges. There have been studies showing that Sebasti's rockfish, the juveniles, will use these glass sponges. You'll find them in higher numbers because they use them for kind of protection and hiding from other bigger fish that want to eat them.
So question for ROV pilots, is this the sort of feature cluster that we might be able to pirouette around?
Sure. Pirouetting now.
Or 3D imaging?
Yeah, it wouldn't take long regardless, so.
Ooh, I would love to see some, we're kind of trying to test this out. So this seems like a nice sort of discrete area that has a nice concentration of sponge morphologies. If you think it's possible, it might be worth 10 or 15 minutes that it would take to approach from every angle, basically.
Nicole, do you have
What do
your
you think,
clicking
Abby?
finger ready?
Yeah, I think that sounds fab. Does it matter if you've got fish sailing in and out? Does that make the reconstruction
Well, they're not
difficult?
going to be helpful, but we'll have to work around them. They might end up being a smear in the imagery.
Yeah.
It kind of sticks out a bit so we can get, both sides here. So it's a good one.
Beautiful. Yeah, as much perspective as you can.
Fantastic.
Thanks, Lizzie, for noting that on the side chat. We'll try it.
Let me know if I'm pirouetting too fast.
I think Chad King is also on side chat, and he is the person who manipulates the imagery with the Agisoft software.
We had success with a sponge of these sponges of these sizes with another different program called Colmap, but yeah, as far as stitching around, even if it's not a full 360, I think you should be able to get a decent amount. The hardest part will be top down and the up and the bottom up. I think Dan will take care of that.
We might go back and look at the other sponge and let it clear up a bit here. It's dusty from the first pirouette.
Sounds good.
You pan down a bit, Michael.
You want me to come down?
No, pan down.
And down.
Maybe slide Argus off
Roger.
to the
To the east of it, just a hair.
to the northeast a bit, maybe.
Yeah.
Get it out behind us there, so I'm not fighting a tether.
Yep. Bridge now.
Oh.
10 meters, 045.
Come up 5, Mike.
Yeah, yeah.
Got a fighter. The fish don't make it easy for Marley having to change the iris either.
Yeah. It's all right, I'm on it.
It's probably going to be a on-the-fly zoom here. You want to zoom in there, you can. Oh, we still got clear water.
It looks like a lot of them around here have this white band situation.
Yeah, it traveled from one to the next, right?
Yeah, I think it's nice to get some good pictures of it because we can send it off to people who work with sponge disease. Might be able to give some insight as to what could be causing it.
Over the past few years, we have seen diseases sort of bloom across the West Coast, notably the sea star wasting disease in the intertidal zone. But I think I'm for sure less aware of deep sea diseases that may be spreading.
Yeah, and this might just be part of the life cycle of this particular sponge, and maybe it's just it's not necessarily
Try
a
a
disease. It
full
could
zoom
be
there
a
if you
change in
want. Or zoom
sediment.
in a bit more on that.
Yep, roger.
Food or oxygen.
Go.
Lovely zoom, thank you.
Got it? Okay.
This is the sort of thing that I wish we had the ability to follow through time to see the progression.
Yeah.
Thank you, ROV pilots and video. That's just what we needed to see.
That's what we're here for.
Avalanche.
Do you think the silt has settled enough on the other side?
It was till that avalanche came down the hill there.
Uh-oh.
That wasn't even you.
We might find another spot with some heterochrome that's not so high relief.
I also got
Is that a
the,
Rex?
I took the imaging when we were passing it the first time and in the reverse direction, so you might
Okay.
have enough images on that one.